Right, so I've been working with a lot of different companies that are trying to bring new therapies for obesity and diabetes to patients. The list is up here. I don't think any of this is actually terribly relevant to the conversation we're going to have. All right, so I'm going to talk to you a little bit about obesity to start off with. Right, we all know that, I don't have to tell this audience about the difficulty of the obesity crisis and its the impact of that epidemic on us as a society. But I think we all know in this room that people's views of that crisis are awfully skewed. We know that if we take in the same number of calories as we burn, we're not going to change our weight. If we're getting more obese as a society, it must be the case that our intake is exceeding our expenditure. The vast majority of people that I interact with, including physicians, look at that imbalance as something like that. Right? But everybody in this room knows that that's really not an accurate picture about what's going on in terms of obesity. Right? So I'm going to walk you through some numbers just so we're all on the same page. Right? So if you're an average-sized male like myself, you're going to consume 900,000 calories in a year. And no, you didn't consume all 900,000 at that buffet. Right? But what's interesting about that is that means that, of course, if we're in energy balance, we're going to consume 900,000, we're going to burn 900,000. So what does it take if you wanted to gain weight, right? How much more do you really have to eat? And the answer is, if you wanted to gain, let's do an analysis, we're going to just gain 1 pound. Now 1 pound doesn't sound like very much, but as bad as those maps of the US that you see from the CDC are, we're not gaining on average much more than 1 pound per person per year, right? So 1 pound, right, really means that you're overeating by about 4,000 calories in a year. Now think of that as an error term. How far off are we in terms of being able to match our intake to expenditure most of the time? And the answer is it's something like 0.4%. Even worse, do the math backwards and say, well, how many calories is that a day that we're within in terms of matching our intake to our expenditure? And it's 11 calories a day. Right? It's a potato chip. Right? Now, a good thing is that you got me after lunch. Right? Well, one time I was speaking to a group of physicians, and they had me go on right before lunch. Always a mistake. And then, you know, I'd use this potato chip analogy, and they had potato chips at the lunch. And I walked out, and one of the physicians had taken the bag of potato chips and dumped it all over his tray. And I said, well, what are you trying to do? I said, I'm trying to figure out which one is the extra potato chip. I hadn't completely got my message through to that particular individual. The point I'm trying to make is that we all want to believe that we are in conscious control of when we put the fork down. But the fact of the matter is that most individuals, including most obese individuals, are within 11 calories a day of matching their intake to their expenditure. And that implies that there is a lot of biological process going on. That is, if we were trying to weigh our food, use a pedometer, all these other things, we couldn't get to the accuracy of 11 calories a day, right? The crumbs that you'd drop, the extra cookie you'd grab when you wandered through the kitchen that you didn't write down in the diary, all these things would be way more than the 11 calories that we really are off in terms of the biology. Okay? And how do you do that? Well, if you think about your adipose tissue as the excess calories, and think of it as a bathtub, well, it must be the case that if we're trying to keep the amount of water in the bathtub the same, it must be the case that the rate of the water coming in and the rate of the water coming out have to match. No difficulty here. Now, the important part about this is there is only one organ in our body that is in the position to be able to regulate both the rate of the water coming in and the rate of the water coming out, and that's the central nervous system. So I've already had a conversation today about the brain initiative. If you ask me about what's the most important organ system when we're thinking about what's going on in terms of obesity, it's not adipose tissue itself, it's not the GI tract, it's the brain. Right? That you have to be changing the brain in order to be able to change the amount of water that's ultimately in the bathtub. Now, in order to be able to keep track of all this, the brain has to have information about how much water is actually in the bathtub so that it can alter both the rate of the water coming in or the rate of the water coming out. And I think everybody in this audience is clear that the way the brain knows about this is largely through hormones, and the most important of those hormones is a hormone called leptin. Leptin's made in adipose tissue, circulates in proportion to how much water is in the bathtub, and informs the central nervous system via receptors about what's going on. Why is it hard to lose weight? Well, when the water levels start going down, right, the levels of leptin fall, the brain responds to that by trying to increase the rate of the water coming in, right, makes you hungrier, and it actively decreases the rate of the water going out. That is, it makes you more efficient at burning calories. So you have to burn less calories to do the same work, okay? So this is what we're fighting all the time in terms of the biology. Leptin's really important, and it becomes very obvious as to why. So one of the important things that you should get out of my talk today is it's not about how much water it is, it's about the signals like leptin. So for instance, this is a normal mouse. If you look at a mouse that doesn't make any leptin, it looks like that. And you can tell this mouse doesn't make any leptin because he can't see its feet, right? And the important point is that animal has plenty of water in its bathtub, but it doesn't know that it has water in its bathtub and goes on eating just as if it doesn't have any water in the bathtub. And again, this isn't just fancy mouse tricks, right? If you find individuals who lack leptin, right, small number of them, but they're clearly obese, right? So this is a 3-year-old that weighs 42 kilograms. If you give it back the hormone that it's missing, right, this individual now comes out to be a relatively normal weight. So again, this is not biology that's just rodent specific, right? These kinds of weight and behavioral determinations matter for humans as well. Now, the question I want to try to focus on is if this biology of body weight is so accurate like I'm trying to preach to you, why is it that we are becoming obese? Right? Why is it that the system that protects us from losing weight doesn't also protect us from weight gain under different circumstances? Nobody here would doubt that one of the large hypotheses here has to do with the food that we eat, right? That the food that we eat has an impact on what it is that we weigh. Not just in quantity, that's obvious, right? But also what the food is made up of. But all of us would also agree there's a lot of confusion about what elements of the food matter to that outcome. And again, we talked earlier, there's conversations about the public communication of this, and it has become, I think, is extremely confusing for the public about what it is that matters. So I go back to some rodent data to make this a little bit simpler, right? So if you compare a group of mice, in this case, that are on a low-fat diet, or a diet, in this case, that's carefully matched for both substance and quantity, except for the fact that it has more fat and less carbohydrate. In the case, the fat here happens to be butter oil. What you see is that the animals that are on the high-fat diet gain more weight than the animals that are on the low-fat diet. And it's not magic about how they do this, right? It's not biochemical magic. They eat more of it, right? They spontaneously choose to eat more of the high-fat diet than they do of the low-fat diet. And the question you have to ask yourself is why, right? What we know happens is that we're increasing the amount of water in the bathtub, and that ends up increasing the amount of circulating leptin. And what you'd expect, of course, is that with more water in the bathtub, the brain should respond to that and reduce the rate of the water coming in or increase the rate of the water coming out in order to bring the water levels back to the same place they were before. But that doesn't happen. And part of the reason is because what we eat matters to the ability of leptin to drive a signal into the brain. I'm going to give you a simple example of that. This is our own version of this. This is an experiment that's been done hundreds of times. Okay? So if you give an animal, this happens to be a rat, an injection of leptin, it eats less, right? Thinks there's more water in the bathtub, I'm going to reduce the rate of bringing more water in. If you take same set of animals, feed them a high-fat diet, give them exactly the same dose of leptin, they're entirely blind to the ability of leptin to do that, right? That's true whether you look at food intake or if you look at body weight as the outcome. The point is that what the animal was eating mattered directly to the ability of that signal to be able to drive a CNS process. So again, what you imagine here is that what we eat can produce something that you might call leptin resistance, that the inability of leptin to drive that same response that it would before. And a lot of my time is spent in trying to understand the molecular underpinnings that connect what we eat to this change in these CNS processes. Right? Now, I'm going to try to propose to you a hypothesis about one kind of molecular link. about how we might connect what comes in through the diet and what happens in the central nervous system as it relates to these systems. And that's going to focus on a transcription factor called PPAR gamma. But the important point here is that when we make an animal obese by access and changing the diet, what we have not done is change the nature of the regulation. All we've done is change where the animal is defending it. And again, this is an important point that I don't think always comes across in the public conversation about obesity. Obese individuals, just because they have more fat, does not mean that it's easier for them to lose it than a lean individual. They are defending it using the same mechanisms as a lean individual does. They are just defending it at a higher point. And that's clearly what happens when we expose these animals to these high-fat diets. They actively defend a higher level of weight. And I'm going to try to make a case to you that this particular transcription factor and receptor is an important part of it. So what is PPAR gamma? It's a nuclear receptor that controls a wide variety of target gene transcription. Okay? The endogenous ligands for PPAR gamma, and again, a lot of this control we know in the periphery relates to adipogenesis and lipid and glucose metabolism, and this will be obvious in a minute. But we know that the endogenous ligands for PPAR gamma are fatty acids and fat metabolites. So what is PPAR gamma? It is, again, that binds to this and then can drive this transcriptional machinery. Its a physiological role for PPAR gamma is to act as a general sensor of the flux of fatty acids in the cell. Right? And again, this is different than when we're talking about, earlier we've been talking about chemicals that come along in the food. The food itself, right, drives this receptor machinery and the biology that's downstream of it. Right? Now, how did we find this? We didn't find PPAR gamma by actually looking for things that responded to nutrients. Right? This was actually found because it's the target for this class of drugs, TZDs, that were used as effective pharmacotherapy for diabetes. These TZD drugs actually are much more potent ligands than anything we've been able to find in nature to be able to turn on this transcriptional machinery, which includes, again, increasing adipogenesis and regulating both lipid and glucose levels. Now, these drugs are great until they've been removed from the market for cardiovascular risk issues. They are successful in lowering glucose, however. The problem comes along is that they're also associated with potent weight gain. So this is human data showing you weight gain on rosiglitazone. This happens to be rodent, just to show you that this is a phenomenon that cuts across species. And the problem with this is that if you went into almost any endocrinologist's office and said, why do these drugs increase body weight? What they'll tell you is it's because it induces adipogenesis, right? That we make more fat cells. It's true that we make more fat cells on these drugs. But as everybody here can appreciate, that if you don't change the nature of the intake or expenditure, it doesn't matter how many fat cells you have, you don't fill them up, right? So from my point of view, as a neuroscientist, that explanation for why people gain weight when you turn on this particular receptor made no sense. So the point is that it almost has to be the case that something is going on to change this expenditure versus intake equation when you give people these drugs, in addition to the idea that we're adding fat cells. And what became very clear is that when you look carefully, this happens to be rat, when we give these drugs, even in the first 24 hours, the animals are increasing their food intake. There's not a lot of new adipocytes that came on in that first 24 hours. Right? So something else has to be going on. And again, the point here is that there's got to be targets for these drugs in the central nervous system as well. Okay? So how do you go about testing that? So what we did was to look to give tiny doses of, in this case, a PPAR gamma agonist, rosiglitazone, into the brain and see if we could change food intake, body weight, and body fat. And what you see here is a dose effect curve. So this is a tiny dose, 1 microgram of rosiglitazone given to the central nervous system increases food intake by 50% over the first 24 hours. One dose, just if you look at it over 3 days, increases food intake for a full 3 days. If you look at the animal's body fat at 7 days, it's higher in animals where we have just tickled these PPAR gamma receptors directly in the central nervous system. Again, very potent effect to be able to drive the response by acting on these receptors. How about if we block these receptors? Right? Is there an endogenous role for PPAR gamma in order to matter? And let me just point out that if you give peripheral rosiglitazone, is it the case that part of what drives it has to do with the ability to drive those receptors in the brain? So again, the experiment here is to give peripheral rosiglitazone and then block these receptors in the brain. I'm not going to bore you with all the different ways we did this, but here's just one example. So again, here's this nice effect for rosiglitazone to increase food intake in the first 24 hours. If we block PPAR gamma receptors only in the brain, you can get rid of that effect. So again, the drug effect does have something to do with being able to drive this. And again, we talked about replicability today. This actually was a paper that was published alongside ours from Jerry Olewski's lab, showing exactly the same kind of thing, using a different approach. All right. So does PPAR gamma matter under normal circumstances, right? What happens when we block PPAR gamma? Can we actually reduce food intake and body weight? And what's interesting about this is if you look under chow conditions when we block those receptors, that is when again, for those of you who don't spend your time thinking about rodent diets, right? Rodent chow would make any vegan proud in terms of how much fat is in it, right? It's incredibly lean. It's about 6% of the calories come from fat. And under that condition, blocking the central receptors doesn't matter. But if you now put the animal on a diet that's high in fat and presumably high in the ligands for PPAR gamma, well, all of a sudden now when we block those receptors, we reduce the animal's consumption of that diet. So again, there are circumstances under which this receptor is activated and that we can show that in fact that it matters to the behavior by blocking that receptor. All right, now we go back to the issue about leptin insensitivity and leptin resistance. Is PPAR gamma part of how a high-fat diet leads the brain to be less responsive to what's going on? So again, as I told you before, leptin under normal conditions will cause reductions in food intake and body weight. If we feed an animal a high-fat diet, we get increases in adipose tissue expansion, increases in leptin amount, and decreased ability of leptin to drive that response. If we now block PPAR gamma in the brain, will we be able to restore that normal response to leptin? Okay. Here is animals now all are going to be fed a high-fat diet. These are animals that get vehicle. Here's an animal that gets leptin. And again, you can see if on a high-fat diet, leptin doesn't do its job. It can't cause a change in the amount of water in the bathtub. But if you now block these PPAR gamma receptors in the brain, we can restore that ability of leptin to drive that response. And again, that's true whether we look at food intake or we look at body weight. So my point here is that PPAR gamma is a receptor for nutrients that links what the animal was consuming to changes in the biology and the signals that are necessary for normal body weight regulation. Again, it's not magic about how it is that the food does this, right? It doesn't have to be some ethereal process, right? It is a process of that the food can drive receptor systems in the central nervous system that are related to the control of food intake and body weight. It uses the biology that we already have that controls our weight in order to drive that response. Now, if you can accept the idea that receptors for nutrients matter to these kinds of responses, it doesn't have to be that these receptors are only involved in food intake regulation. They're likely to be involved in lots of other aspects of biology. And one of the ones, I won't bore you with exactly the rationale here, but that we got interested in was the idea that various aspects of stress responses were impacted by PPAR gamma. And let me show you a couple experiments that actually test that. And in particular, we're interested in this because we know that there's issues about whether the changes in our environment, right, that relate to psychological stress have an impact on these systems that matter to our food intake and body weight. Okay, so here's the experiment. We're going to take a rat in this case and put it in a small container. As you can imagine, that makes the animal unhappy and stressed out. Right? If we put you in a small container, you'd be unhappy as well. Right? We do this for 20 minutes, and what you see, this actually happens to be heart rate. You can see that their heart rates shoot up by almost 200 beats a minute when you do this. If you treat the animal with PPAR gamma agonists, that response is greatly diminished. We reduce that response to that stressful event. It's not just these kinds of sympathetic changes, but it's also the case that endocrine responses change, right? So we all know that when we put these animals in these restraints, that we get increases in glucocorticoids. Again, that response is greatly blunted if we treat the animal with PPAR gamma agonists. Why do I bring this up? I bring this up because we have to ask ourselves a little bit about why it is that animals would choose foods that have high PPAR gamma ligands in them, right? That then sets off this cascade that ends up in higher weight. Well, part of the answer could be that we are actually changing stress responses as well. And in some ways, when we talk about the term comfort food, this is a little bit about what we're talking about, right? The idea that some foods may drive biological responses that people find helpful in terms of their dealing with whatever is going on in their lives. Now, I'm not trying to say that this is the only way we get there, but it can at least be part of trying to understand the relationship between types of foods and outcomes. Okay, why am I bothering you with all of this? I'm bothering you with all of this because I try to use this PPAR gamma system as a single example about how we can think about food in a way that's different than what at least most people think about food. I don't think it's going to be terribly novel to this audience to think about food as driving receptor systems in addition to driving other aspects of the biology. But the reality is that most of our dietary recommendations come from two sources of information. One is the idea of the biochemistry of food, how we break it down and turn it into ATPs. And we've already had some conversation about this around the fructose argument, right? The idea that fructose is negative starts with a biochemical argument about how fructose is different than other sugars. The second way we come up with dietary recommendations has a lot to do with nutritional epidemiology, where we break down various aspects of what we can measure about what people are consuming and correlate it with different outcomes around health. Now, I'm not here to say anything like these are not appropriate approaches. All I'm saying is that, again, we talked about the difficulties of making clean, the replicability around some of these conclusions that come from these have been difficult. And I would say it's not just the fact that there are genetic variations across the population, but it's also the case that there are other aspects of the food that are harder to delineate at a nutritional epidemiological level. So food has signaling properties. We call them, we've made the analogy to a hormone, right? That it's introduced into the body, acts on distal sites in the receptors. In the case I've just shown you, PPAR gamma acting in the central nervous system. But this isn't the only example of this, right? There are lots of other examples where it's clear that some of the biological effects that explain how one source of nutrients differ from another are not just about what their biochemical properties is, as how we get them to ATPs, but are about these other aspects of the biological systems that they drive. And my point is that a complete idea of how we actually use diet to help people achieve different kinds of health outcomes can be built, some from the top down by looking at what people eat and what happens to them when they eat it, but can also be built from the bottom up. That is, if we're trying to turn on X, Y, and Z signaling systems and turn off A, B, and C's, well, we are learning more about how to do this every single day in terms of what types of nutrients will do that. And that the design of diets are going to have to take into account the idea that we can drive these parts of the biology in addition to the information we get from these two other approaches. So I will stop there and point out the obvious, which is all the work you just saw wasn't done by me. It was done by a talented group of folks and collaborators that I have at the University of Cincinnati. This is Steve Woods, my longtime collaborator and friend, who I know has been part of this organization in the past. This is Karen Ryan, who led much of the work around PPAR gamma in the brain as well. And I will thank you for indulging me with this opportunity to come and talk to you today, and I am happy to take some questions if you'd like. So thank you very much.